I'm Dr. Shakti, and we are going to start with physiology today. So we already done with the foundation, orientation, and discussion of schedule. So today onwards, we're going to start with general physiology for the next three classes. So we'll be talking about general physiology today. Okay. So just before that, just look at few questions that usually come from general physiology. Like, for example, feedback regulation, what is negative feedback? Okay, what are the intercellular connections? Transport is another thing. Okay, again, homeostasis, cell membrane apoptosis. So this is a large chapter. We'll be dividing it into three sections. So largely today we'll be talking mainly about your cell membrane. Okay, so one class I'll keep it for the cell membrane and its functions. I'll keep a separate class just for transport, which I'll be taking on the third day. And the rest of the things, I'll be doing it in the second class. Okay, so this is based on how significant each of these topics. That is how I'm allotting the topics for each of these things. Okay, so I'll just start with the lecture for now. Yeah, so later once that is done, we'll discuss more about what Auricle is and how the features of Auricle are. Okay, so let's talk about your membrane. So I'm just putting in, three beakers right over here like this. We're going to fill it with water. Okay, once that we've filled it with water, I'll take a substance like a phospholipid, for example. So phospholipid, phospho and lipid. So it has a phosphate group and a lipid layer. So it's going to look, something like this. This is just a very basic, I'll describe it more in detail. So it has one head part, which has a phosphate group, and it has a fatty acid chain over here like that. Okay, so this head part, which is phosphate rich, that is polar hydrophilic. On the other hand, the fatty acid containing component is hydrophobic. And this part is polar and it is hydrophilic. So please tell me, when I put it into a beaker of water, okay, and there is an air-water interface, what is likely gonna happen? It's gonna line up in a way that, can you see the polar end? That those polar ends are gonna touch the water, okay, while the one which is hydrophobic will go outside. So it's all about energy conservation. Out of all the possible ways this can align, this is the most energetically favorable way this can align. Okay, so this is one way it can happen. Now, what we're going to do is we're going to increase the amount of your phospholipid. Once we increase the amount of phospholipid, you start seeing that instead of this, it's going to slightly differ in how it's going to be put. So the water is there, and away from the water, your hydrophobic ends will come in the center. So this sort of an arrangement will be there. So this is a mono layer. And this is what you call a micelle layer. Now, if we further increase the concentration, if a good amount of phospholipids are there, what is going to happen is it is now going to arrange itself in a bilayer fashion. So what is going to happen instead? Your phospholipids polar end will be touching water, and the hydrophobic fatty acid will be put right in the center. So this is a bilayer formation. So in physiology, in your body, actually all three of them are present, if you think about it. So this monolayer, for example, is present in your surfactant layer, in the alveoli. If you look at the micelle layer, in your GIT, in the intestine, with regard to, bile acids and all, and how it helps in digestion. So this is one way that is there. This bilayer is a classical example of how your cell membrane is existing. So all of these ways, based on the amount of concentration, what is the nature of the fluid, how it is shaped, based on all of these criteria, these will be present in your body. Okay. So this is basically your bilayer structure. So through this bilayer structure, what happened? What you can understand is a compartmentalization happened. Okay, a compartmentalization happened whereby you just created a empty space right in the center, distinct from what is there outside, using this particular phospholipid, which repels the water away. So in this way, what we are able to achieve is we are able to create a sort of a cell-like structure. We are able to create a cell-like structure like this, and we are able to distinguish and differentiate your intracellular fluid from your extracellular fluid. And for that, what is the requirement? We have connected to the extracellular fluid a polar head, connected to the intracellular fluid a polar head, and a phospholipid layer right in the center like this. So through that hydrophobic layer, it is separating these two compartments. And this extracellular fluid can further be divided into two compartments. That is your interstitial fluid and your plasma. So consider there is, a blood vessel which is coming from here. Inside the blood vessel, the fluid which is there, that is your plasma. Okay. And cell, between the cells, between the plasma and the cell, this small space is where interstitial fluid is there. These both together is what makes up your extracellular fluid. And then, of course, you have the intercellular fluid. So this is a way of compartmentalization. Membrane is very important in order to achieve this one thing. Okay, so it's not only the phospholipid that is there. So it is a kind of lipid. Apart from lipid, we also have other constituents. Like, for example, a protein structure. Okay, and some of these protein structures, if you notice, can sometimes have a carbohydrate moiety on it. So in your membrane, you have your lipid, you have your protein, you have your carbohydrate. The percentage of all of this varies according to the type of cells. Most of the cells that you see, protein is going to be much higher. Not much higher, just slightly higher. Okay, so if you look at the different kinds of cell, one example is, nerve cells that contain myelin. So myelinated cells generally have higher lipid than protein. This is for myelinated cells. Otherwise, most cells almost similar with slightly higher protein. Most of the other cells. Now, one type of cell where protein is much more than your lipid, much more than lipid, that is in your mitochondria. So mitochondria has your protein more than your lipid. All right. So you have your concentration percentage. So some MCQs can come from this. Myelinated cells generally have higher lipid to protein. On the other hand, mitochondria has more protein to lipid. Otherwise, most of the cells have, 50-50 percentage of lipid, carbohydrate, protein. Out of that 50 percentage comes to around protein, but slightly higher. Some textbooks say give a value of, just for example, I'm just giving you a reference value, 55% of this. Okay. But again, as I'm telling you, there is a lot of difference based on the type of cell that we're talking about over here. So now let's go into the detail. So short note that can come is What is cell membrane? What are the parts or constituents, composition of cell membrane? Separately question of, lipid structures or describe about phospholipid. Or you can talk about what are the protein structures, okay, and functions of carbohydrate structures. So in various ways, we can talk about your lipid, protein, carbohydrate, or in general, you can talk about what are the functions of your carbohydrate or your membrane in general. Okay. Okay. So first, let's start off with lipids since we already started with lipids. That is a good way to start off to understand your membrane. When you talk about lipids, the most common example is your glycerol based. So glycerol based phospholipid is the most common example. Okay, so these phospholipids, what is the structure of it? Why is it glycerol based? For that, you have to understand that one, it has a glycerol component. Okay, now attached to the glycerol component, you have your fatty acid chains. Usually they have two fatty acid chains attached to it. So one fatty acid chain here, another fatty acid chain here. On top of it, there is a phosphate group which is here. Okay, so it's just going to be like O, 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 O like that. Just for your understanding. So we have a glycerol, fatty acid chains, and a phosphate group. And usually on top of a phosphate group, there is going to be a particular head group. So we got a phosphate, we have a glycerol, we have two fatty acid chains and a head group. Now this head group can vary. There are different types of head groups. It can be a ethanolamine. It can be a serine. It can be a inositol. It can be a choline. So based on this, you can call it in different names. Phosphatidyl ethanolamine, phosphatidyl serine, phosphatidyl inositol, phosphatidyl choline. These are the types of phospholipids which are commonly seen. Now, usually this choline, phosphatidyl choline, is usually present on the outer leaflet. And this is usually present on the inner leaflet. What does that mean? As we were talking about this, we have the outer leaflet, we have the inner leaflet. Generally, your phosphatidylcholine is present in the outer leaflet. Things like ethanolamine, inositol are all present on the inner leaflet. This is important regard to its functions. So phosphatidylcholine, for example, is one area whereby certain things can attach to it from outside. Okay, so in platelet aggregation, after that addition, in that step and coagulation cascade, this is important. Phosphatidyl inositol, which is there on the inner leaflet. That is very important when you talk about signal transduction. In a lot of ways, this phosphatidyl inositol gets split into two components, and so further signal transduction pathways can take place. So as I'm saying, these are limited mostly to either the outer or the inner leaflet of your membrane. And there is something called a flip-flop mechanism also, which can happen, which is quite slow actually. Okay, just putting it out to you. So we have a head group, we have a phosphate group, we have a glycerol group, and we have a fatty acid chain. Now here are some important points regarding this. So this fatty acid chain, based on the length of the fatty acid chain, one, and based on whether there are double bonds. When there are double bonds, what will happen is it will start kinking and turning to one side like this. So presence of double bonds is one thing, and the length is another thing. So if the length is increasing, okay, and no double bonds, That is, it is saturated. No or less double bonds. Then what will happen? It will be a long thing, okay, without any kinking. So there is higher chance for interaction between these tails. Okay, so at rest, what you have to understand is the interaction energy is pretty high. Okay, so generally you have to understand your membrane. Then there is a particular concept about your membrane that you have to understand, which is called fluid mosaic model. What is fluid mosaic model? So this structure of your membrane is existing in a sort of a fluid form where it's constantly shifting. There's flip-flops, horizontal shifts. All right, so a lot of shifting is taking place. And this property that we're talking about depends on temperature, for example. Okay, so your membrane can either exist in a gel form or a sol form. And when you increase the temperature at one particular temperature, which is called the transition temperature, what will happen? This gel form gets converted to sol form. Okay. So if the interaction energy is high, in that it has longer length or saturated areas, then it requires a higher temperature. So transition temperature will be higher. On the other hand, if it is a shorter length of tail of fatty acid, it has more double bonds present in it, then the transition temperature is going to be lower. So because of this, there can actually exist different domains within the same membrane. Some areas where, for example, longer fatty acid chains are there, more saturation is there, then there is, at a particular temperature, it can exist in more of a gel state. And if you go to some other part, it can exist in a more solid form. So such a difference where micro domains of different fluidity are also present. Okay, so this is all the important concepts regarding your phospholipids. Okay, so I'm trying to teach you things more than what you must have studied in your, school and all that. All right. So first thing we did with lipids as a glycerol-based phospholipid and how it is helping over here. Okay. Now beyond this, just try revising the structure again, because based on this structure, similar structure is also what we're going to discuss now. So here, what is there? Glycerol is there. Two fatty acid chains are there, which are the tails. A phosphate group is there. Attached to it, a head part is there. Now, apart from this, the next type of lipid is your sphingolipid. So these sphingolipids, there are different types. Instead of glycerol, there is one particular part which is called sphingosine. So this sphingosine, it is in such a way that it by itself has a carbon chain like this. And on top of that, of course, a separate fatty acid chain like before will attach to it. Now to this, you attach a phosphate group and again head, very similar to what we are talking about. Only thing is replace this glycerol and one fatty acid chain and make it a sphingosine molecule. Okay, so this structure is what we call a sphingomyelin. Now, instead of this, we again take a sphingosine, fatty acid chain over here, And to this, we directly attach any sugar residue. So here I'm just going to put a galactose. Okay, so this is an example of what we call galactocerebrosidase. This is a glyco, because a glucose like moiety is the sugar component is the glycosphingolipid. Lipid sphingol with a glyco component. So this is a glycosphingolipid. One example is this. Okay. Now you take any, glycosphingolipid. To a glycosphingolipid, you attach a sialic acid. You attach a sialic acid. And that will give you something else called a ganglioside. So what do you have? A sphingolipid. So first thing was a glycerol-based phospholipid. Then we have a sphingolipid. That sphingolipid can be divided into a sphingomyelin, a glycosphingolipid. One example is galactocerebroside, and there is also a ganglioside. Now with diagram, you can draw and explain all of this. So one of the thing is, students, when these questions are asked, they do not have enough points to write all of this. Okay, with diagrams now, you have plenty of points to write about them. So now what exactly does a sphingolipid do? You understand what a phospholipid does? Sphingolipid in a lot of ways is important in cell to cell interaction. It has some receptor role. Okay, so in a lot of ways, cell to cell interaction, it has some sort of a receptor role. And, sphingomyelin along with cholesterol form certain microdomains called the RAFs, which are again important in certain kind of intracellular signaling, like caveolin mediated mechanism. Also, the sphingomyelin, by the presence of a particular enzyme, sphingomyelinase, which is usually present in the lysosome, that can bring out ceramide components. So ceramide components, has some role in helping in invagination of your cell membrane. There is a particular disease, a lysosomal a storage disease, where this lysosome does not have sphingomyelin. It is called Niemann-Patrick disease. Okay, so there's a particular disease called Niemann-Patrick disease, which happens when the sphingomyelin is not present inside the lysosome. Again, as I was telling you, this in general, cell-to-cell interaction, receptor role. Apart from that, some extra things with your sphingomyelin functions. Okay, so these sphingomyelins are also, found in your myelin and your parts of your lipid transport mechanisms, actually, lipoprotein when you talk about. Okay, a lot of the things I might be talking about might not make sense. Write it down, please, because as we keep going through, you'll hear these terms many times throughout physiology, through biochemistry, at which point, once you start jotting them down, they'll start making more sense to you. Okay, so we talked about one type of lipid, that is glycerol-based phospholipid. Two, sphingolipid. The third category that we have to talk about is your cholesterol. So cholesterol has an area like this, which is basically a sterol ring. On top of that, it has some carbon chain also. So it has two things. One, a sterol nucleus area and some carbon atom chains. Okay, so if you look at it in a way, it's actually very similar to how your other lipids that are involved over here are. In that, it has one nucleus-like area, which is going to be a polar area, and the hydrophobic area. So this also has a similar activity. But this is very important in your fluidity, actually. Fluidity of your membrane, your cholesterol is very important. So this depends on two factors. One, the concentration of the cholesterol. Two, the temperature. The thing is, usually when temperature increases, the fluidity of a membrane increases. When temperature decreases, the fluidity decreases. Okay. What cholesterol does is, it prevents, excess fluidity when temperature goes overboard, and it prevents freezing when temperature drops. So when temperature is high, it decreases fluidity. When temperature is less, it increases fluidity. On the other hand, when you're talking about the concentration, if it is in, some modest concentration levels, it actually decreases fluidity. But when it is in high concentration, it increases fluidity. So explaining all this is beyond the scope of just 1 hour. So if you have any doubts, please do let me know. If you want me to explain anything a little more extra as to why this is happening. Okay, you can contact me in any way or in future sessions. More than happy to help you out with this. Okay, so we have here glycerol-based phospholipid, sphingolipid, cholesterol. What do all of these things do? That is essentially your lipids. Now that we're done with lipids, we'll go to the next structure that we're talking about, which is protein, which is pretty important. When we talk about protein, two things I want to put into your head. There are what we call integral proteins, and then we have what are called the peripheral proteins. Now, students don't really understand what exactly this word means. Transmembrane integral not interchangeably used. Okay, the thing is, one type of integral is a transmembrane protein. So integral proteins are covalently linked to your membrane structures, your phospholipids, for example. So these proteins, which are integrally linked covalently, they are integral proteins. So in order to, in experiments, if you have to bring out the integral proteins, you have to actually destroy the membrane through detergents and all. But peripheral proteins, just by, tweaking the temperature, salt concentration, all of that, you can bring out peripheral proteins without damaging the membrane. But integral proteins, in order to bring it out, you need to destroy the membrane through use of detergents or something like that. Okay, so that is the basic difference between what an integral protein is, what is a peripheral protein. This integral protein can be of few different types. Again, this is a separate short note that comes. Please do remember. Okay, so don't sleep on this. Protein and its functions are separate important question that comes. Okay, so one type is when you look at the membrane, there is a type where it spans throughout multiple times. Like, for example, 7 types. Serpentine transmembrane proteins, which has a N terminus and a COH terminus. Okay, so this is one type of thing. Okay, just to make it a little more beautiful for you to understand it properly, on 2, 3, 4, 5, 6, 7. Okay, I hope you are able to understand it a little better now. This is actually 7. So these are all, linked to each other like this, like this, like this, and they span through the membrane 7 times. So this is one example. This is a transmembrane protein. There can be other proteins where they might not necessarily span through multiple times. They can, span through only single time. Uh, they might not span completely, but are covalently attached. So they still are under the category of integral proteins. Okay. There's another variety, which is what I call GPI anchored. So what will happen is the phospholipid which is there, some component of that phospholipid attached to it, another phospholipid, attached to it, some sugar molecule, attached to it is a protein. So this protein is outside. Usually people think that, what is outside is peripheral, but this is also covalently attached and will come under integral proteins action. So this is usually this is what we call GPI. Okay, glycosylated phosphatidyl inositol. Remember the phosphatidyl inositol to that glycosylated, a protein which is attached. Sometimes, so this is usually there in the outer leaflet. Apart from that, in the inner leaflet, certain components which are attached instead of, phosphatidyl inositol, it can attach itself to myristoyl, prenyl, different groups like this. Okay, so one example for this is what you call SRC. This is the RAS. None of this is going to make sense. So these are molecules you'll come across in your endocrine system when insulin, for example, acts. Okay, so these are integral in the functioning of signal transduction in our body. So these are examples for inner leaflet integral proteins which are anchored to something else. So these are the types of your integral proteins which are there. Okay. Peripheral proteins, on the other hand, will be non-covalently attached to your integral proteins. Can you see this like this? So attached to this will be another protein like this. It can be inside, it can be outside. It's not like only it's going to be inside. Okay, in this example, I'm showing you one which is inside. Okay, so here again, this is a separate protein which is there, which is inside. Access to these proteins. The first one is what we call ligand receptor. First one is ligand receptor. What is the ligand receptor? So any sort of a ligand which is coming from outside, bringing some sort of information. That information now has to pass through the membrane, go inside, tell the cell, okay, this is the signal that is coming from outside. So how can some substance which is hydrophobic or hydrophilic based on the nature completely pass through? Okay, if it is hydrophobic, it can easily pass through the membrane. If it is hydrophilic, it cannot pass through. So these sort of substances now has to have some sort of receptor outside. So how does this receptor now, after picking up the signal, tell the inside what is going to happen? So it has to go through some sort of a structural change. So in response to some stimuli, like a ligand from outside, these receptors undergo structural changes to communicate through various ways. One of the ways it communicates is, so this is a perfect example of a ligand receptor. Okay, so one of the ways this can communicate is this receptor by itself is a ion channel. So once some ligand is binding, it opens up a ion channel, which is itself or something else. Two, it can through these peripheral proteins which are attached to it. Say this, for example, this activation will cause some changes in this peripheral protein, which will now go and act as what we call second messenger and go on ahead and tell it to somebody else. Or there are ways by which instead of a peripheral protein, there can be an enzyme which is attached to this. So that enzymatic change also, usually a kinase, okay, tyrosine kinase, something like that, that is going to again go and activate your signaling downstream. So that is different ways by which one, these proteins can act as a ligand receptor. Okay, this is first function. Second function is cell adhesion molecules, CAM, cell adhesion molecules. So I'm going to just draw one cell over here. Right next to the cell, I'm going to put another cell. Now I'll just put the bilayer membranes just for your understanding. Okay. So I'm going to put certain integral proteins on either side. So this integral protein which is there, which is kind of jutting out like this. So this integral protein, okay, we'll discuss this detail in next class. For now, I'll just name it cadherin. This is a calcium dependent mechanism. So through the help of calcium, they cross contact with each other. And of course, inside also there are some peripheral proteins. And beyond it, it is attached to certain filaments and all. It can be either an actin or a microfilament or an intermediate filament, for example. Okay, so based on that, the name of it changes. So this is what we call a tight junction. Not tight junction, sorry, adhering junction. Okay, so this is what we call an adhering junction. It's adhering to each other like that. Apart from this, we can also put one here. And in this scenario, say it's gonna, through either actin or a intermediate filament, it is going to combine to some substance in the extracellular matrix. This is what we call a integrin. Okay, so in various ways, cell to cell, cell to basal lamina sort of a connection can take place. And this is very important that one, it helps in preventing metastasis. So in cancer, what happens is when the cells cannot, the tumorous cells which are there, they cannot stay together anymore because of some issues in any of this. They go apart, go through the bloodstream, reach somewhere far away, and start growing there. That is what metastasis means. That should not happen in a normal scenario. That is because of the presence of such cell adhesion molecules. Okay. Apart from that, cell adhesion molecules also helps direct your immune cells to different areas as per the requirements. Okay, your guided axonal growth. Okay. And your resistance to shearing, stress, maintaining epithelial monolayer and integrity. So these are various functions of your cell adhesion molecule. So under the question of what are the functions of proteins, one, you can with diagram explain about your ligand receptor. Two, you can talk about your cell adhesion molecules. Okay. Now, beyond this, we have some sort of cell signaling which is there. Okay. So your before that, I'm just going to go through transport. We'll be discussing this in detail in the next class. But before that, I want you to understand few things about transport. Okay, what is the difference between a pore, a channel, a carrier, and a pump? So these are terms that you'll come across. Students think they're all the same, but they're not, far from it. Okay, so I'm just going to jot down some things that might come as an MCQ or draw the following or something. There are dimeric structures, which are particular type of CIC type of chloride channels. Just note it down. Okay. There are trimeric structures, which is your epithelial sodium channels. This is important for your entrance MCQ as well. Then we have tetrameric, like for example, the potassium channel, your aquaporins. You have pentameric, your acetylcholine, and certain types of your chloride channels, which are GABA, glycine, CFTR. Okay, so these are just examples I'm just giving out for you. So those of you who want to earn more marks, are competitive in exams, try to learn this. And later on, you'll, in fact, require this for your entrance examination. So I'll just draw this one example over here. Tetrameric means 4 subunits. Okay, so we have 1 subunit, 2 subunit, 3 subunit, 4 subunit. This is what it means. Aquaporins also tetrameric, 1 subunit, 2 subunit, 3 subunit, 4 subunit. Basically, this is what it means. Okay, dimeric means this is 1 subunit, 2 subunit. Now, there's a slight difference in how the pores are made. So in potassium channels, which are tetrameric, at the center of this is where you have the pores. On the other hand, if you take a look at your aquapore, right here at the center of each of the subunit, that is where you have the pores. So that isn't of them like this. So they have different subunits. Each subunit is made up of, just like we saw earlier, like this. Okay, multiple time membrane spanning substances, proteins such as like that. There's something called a P loop and all. Okay, and in between them, they can have pores. This is generally for ions, usually. Pores, generally for ions, large structures. Even proteins also can be carried sometimes. Channels, generally channels are just you can draw something like this to understand what channels are, through which your fluid is going to pass through. Okay, so this thing that I'm talking about, this aquaporin channel, that is a good example of a channel action. Okay, so this is actually for your water. not ions. Aquaporin is a good example of a channel. Okay, so the rest of them all are four different ions or some neurotransmitter links. Why is this required is because your cell membrane is having a bilayer with a sandwich of, at the right in the meaty middle of it, we have your lipid structures, fatty acid. So something like water or polar substances that go along with water cannot pass through the membrane. So you need some sort of a protein structure to allow them to pass through. Okay. Now, another question is, if it is supposed to be a substance which is polar or non-polar. Okay, over here, what is there? This area is supposed to be hydrophobic. Okay, if this protein is also hydrophobic, will it allow water to pass through? In the center, it won't. If it was polar, will it stay there? It won't stay there. It'll get pushed out. So how do we solve this problem? Two ways. One, the protein structure can either exist in an alpha helical model or a beta sheet model. So one thing is it can go into a beta sheet. Two, it can go in alpha helical model and is amphipathic. That means it every 3 to 6 amino acid it keeps turning, 3 to 4 amino acid, it keeps turning like this. And because of that, the polar amino acids and non-polar amino acids are alternating between them. So this kind of creates a scenario where, say, if this area and this area is completely hydrophilic. On the other hand, what is there outside this area becomes hydrophobic. So like that, we just solved a major problem. So this is how these pores are able to carry substances through them. Okay, so that is a pore, a channel. What exactly is a carrier? So you have the membrane. Okay, and let me just draw it slightly thinner. Now we have a substance here, high in concentration, but polar. Okay, low here. Now this has to go in. Okay. These pores can be gated, non-gated, but ultimately it goes through it. Sometimes some substances, not just through pores, require some transporter to carry it to the other side. Like for example, let's take example of glucose. It needs a transporter, which will take it here. Okay, usually it takes it along with sodium and then takes it to the other side. So even if, concentration gradient, that doesn't matter. Different types of them. Like for example, fructose, for example. Okay, all this we'll discuss it properly in the third class. That also requires some sort of a transporter, even if it is going down the concentration gradient. Some carrier protein should be there, which will carry it down. Undergoes some conformational change, attaches here, changes, flips, and puts it this side. So that is a carrier. On the other hand, a pump is a substance, a protein substance, which is there on the membrane, which goes against the concentration. From a lesser area, it can go against the concentration and put it somewhere. But since it is going against the concentration gradient, it requires ATP. And it will have an area for ATPase where ATP, energy from ATP, will be used to carry this out. So this is the difference between a pore channel, a carrier, a pump. Okay. Now, apart from this, we have enzymatic activity. Even integral proteins. Usually students write this under only peripheral proteins, but integral proteins also have enzymatic activity. One example is your intestine. Okay, in intestine, we have something called brush borders. It helps increase the surface area for absorption. So say, for example, there is an amino acid and an amino acid. This is what you call a dipeptide. Not a big protein. By the time it's reaching the intestine, it will be, something like this. In this dipeptide, there is in the membrane of these brush borders certain integral proteins which act as enzymes. For example, dipeptidase. So what does it do? It takes this, splits this to give just amino acids, which is now much more easier to absorb into the cell. Okay, so this is one example of dipeptidase. These pumps are also examples. They are using ATP. ATPase is being used. Okay, so in a way, pumps are also examples of an enzymatic activity. Okay, so what do we have here? 1, ligand receptor. 2, cell adhesion molecule. 3, transport. 4, enzymatic activity. All right. Now we'll come to your peripheral proteins. It's somewhat similar in a few things. So one thing is signal transduction. So in signal transduction, usually, I was showing you this one particular diagram, a protein like this. One classical example of a peripheral protein which is attached to it is G protein. You will come across it a lot. Okay, so G protein usually is a protein which has an alpha and a beta and gamma segment. Usually to the alpha, a GDP is attached. And when the signal comes, GDP gets replaced with GTP. And this alpha and beta gamma splits. And it results in a lot of changes, signal transduction like this. So that is one thing that can happen. Second is a enzyme through which this can happen. For example, a peripheral protein which has a Janus kinase activity, which will activate another molecule. So basically, this has some sort of enzymatic activity of tyrosine kinase activity. So tyrosine residues are phosphorylated. Okay, so this is one example. Okay, growth hormone, for example. Now, apart from this, they're also involved in cell adhesion molecules that we were talking about. Beta catenin, plaques, okay, all of these components which are associated with integral proteins like cadherin that I was talking about. Usually next to cadherin, there is a catenin. Next to cadherin, there can be a plaque. Attached to that will be your filaments. Okay. Then a very important thing, submembranous cytoskeleton. The thing is, if you look at the membrane structure, just below the membrane, usually cytoskeleton is there. Cytoskeleton as in, it can be actin, it can be myosin, it can be microtubule, it can be intermediate filament. These are your cytoskeletal structures. A membrane is attached to your submembranous cytoskeleton. Okay, it helps in bringing out the rigidity of the structure, structural integrity. This is very important. One example I'll give you is in RBC. So in RBC, what is happening is there is a particular component over here in the membrane, which is called a band 3. This band 3 is ion channel, okay, exchanger actually, that is attached through a protein. protein 4.2 to a substance called ankyrin, which anchors it to another cytoskeletal structure, which is called spectrin, which is very important. Please remember this. Spectrin, ankyrin, band 3. At your level, that much is enough. Okay. For those of you who want a little more, there is also your actin structures over here. There is your actin structures are also here. That attached to another glycophorin and all. So basically what is happening is all of these actin structures, if you just put it in different ways like this, these are all different actin structures. Now, all of these actin structures through these cytoskeletal structures get interconnected with each other and provides a structural integrity. This is one of the reasons why your RBC, while it is passing through capillaries, squeezing through it, through the spleen, sequestration, all that, it does not rupture. Okay, this is very important. Now, once this is absent in certain genetic conditions, what will happen? This, instead of this, it will become huge and spherical with no shape. Otherwise, what is the shape of RBC? From the sides, if you look at it, it will be like this. Instead, it will now be spherical, which is called hereditary spherocytosis. Cytosis. Okay, so that is with your structures of your protein. The last thing is your carbohydrate. Because of the lack of time, I'll just, rush through it very fast. If you have any doubts, ask me later. Okay. So carbohydrate structures can be a glycoprotein, main structure is protein, which has a glyco component. It can be a proteoglycan. Main is glycan, on top of that protein is there. Or, as we saw earlier, glycolipid. Main is lipid, on top of that. It depends on what is the structure. A big protein with some glycation components, or majorly glycation components with some protein attached to it, or a lipid structure with some glycation on it. What is the function of this? Generally, one example is it gives a negative charge to the surface through some substances like glycocalyx, which is actually outside, not on the membrane per se, it's outside the membrane. One thing. It helps in cell-to-cell interaction. Okay, in terms of one, recognition. Recognition of immune complexes from outside. Also, in terms of adhesion. So there are certain things like E-cadherin, selectins. Okay, these components help in adhesion over here, especially this thing called lectin. Okay, so recognition, adhesion, negatively charged components. There's some bit of receptor activity in some of them, like either for hormonal or immune as well. Okay, now apart from this, there are, based on the glycation. For example, there are voltage gated channels. These voltage gated channels, like potassium channel, KV 1.3. Okay, not 1.3, 3.1. What they do is based on the amount of glycation that is there, the threshold of it can change. How fast it can open, close, it can change. Okay, speed of transmission can change. So glycation, amount of it changes these components. So in a nutshell, if you're asked about membrane, Now we discussed what all is there in the membrane, what are the functions of membrane. So each of them are the functions. Now, if your question of what are the functions of the membrane, you talk about the functions of protein, you talk about the functions of carbohydrate, you talk about the functions of your lipid, you have the functions of your protein. Okay. So why choose Auricle? Because there are recorded classes, live classes like the one you've been attending right now. Practical sessions are there. Also, you have examination modules and hard copy book sets that are made available to you. So again, recorded sessions, pre-recorded sessions, you can look at it at your own leisure and time. Live classes, if you want a one-on-one interactions with a doctor and prefer, attending classes rather than pre-recorded sessions. There is express exam module right before the exam if you want to revise quickly. Okay, everything is within 20 hours completed. There are one-to-one doubt clearing sessions if you require it. Hard copy notes are made available, which has beautiful diagrams and flow charts and questions. There is also a question bank in the app where different degrees of expertise are there from beginner to expert level. Okay, practical sessions are also there. And these are the various subscription plans. You can use my code Dr.Shakthinag 20 for 20% discount on subscription. All right, so I hope to see you again tomorrow, the next session. So thank you for watching. And these are the various social media content that you can contact Team Auricle through. All right, thank you.